Hello. I'm very sorry for this pre-recording. Hopefully, there's no feedback. And whoever gave my introduction, thank you very much. I'm really sad that I couldn't be there. I grew up in the Central Valley of California in Modesto. I was an undergrad and postdoc at UC Davis, and I wish I was there in the sun with you all. But unfortunately, there was an ice storm in Michigan. And so I got grounded, and if everything's going to plan, hopefully I'm flying right now from DC to Dallas, and I'll be there tonight in Davis, hopefully to meet some of you. I'll be talking about topological data analysis today, and you might be wondering why talk about this really specialized field of mathematics when this is about plants. And the reason is because I'm really passionate about quantifying the plant form and plant morphology and architecture. And the ice storm, I think, really highlights plant morphology. When the ice coats all these structures, you really see their contours, shapes, branching architectures, patterns. And when we see aesthetic things and beauty in nature, it's because of these very complicated mathematical relationships. And the reason we pick up on this is because we're all born with this inherent image processing in our own minds. The challenge is how do we quantify this ability we have to find patterns in nature so that we can make a science of it? And that's where topology comes in. And this is my credit slide. And I want to thank a lot of topologists. Mao Lee at the Donald Danforth Plant Science Center was the one who introduced me to topology. And I'm currently working with my colleague Liz Monk at Michigan State University in the Department of Computational Mathematics, Science, and Engineering. And we're working with a graduate student, Mitchell Ethan, who produced a lot of the work that you're going to see today. And mathematicians really love it when the plant biologists follow them on Twitter. They really get a kick out of it. So if any of you in the audience have Twitter, follow Mao right now at MaoLee0923 and Liz Monk at Elizabeth Monk. So why topology? The goal here is to measure global features of shape, the whole thing, quantitatively, and also across scales from the micro to the macro. Imagine if you're looking at the morphology of an organism, we want something that takes in the cellular tissue, organ, and organismal structure all at once. And all of this, of course, are really complicated geometric structures. But before we get too far into topology, I just want to mention that you can do this through other ways, using statistics from a field called morphometrics. And one of the most simple and powerful methods of morphometrics is landmark analysis. And this is really simple. These are just coordinates that are on homologous features that can be found in every one of your samples. And so in this example here of a grapevine leaf, we're placing these 21 different points on every single sample. They're homologous with each other. And then we use an analysis called procrustes to superimpose all these points on each other, minimizing the distance of all points to each other all at once. And then you can just do statistics on your coordinates. And the nice thing about shape analysis is that when you do things like a principal component analysis, you're not working with things that are that abstract. They actually have a form and a shape and a morphology to them. And so when you make eigenrepresentations, for example, of eigenleaves from a PCA, these representations, you can really look at them at a glance and see the shape variance that they're explaining. This is a video I made of a discriminant analysis on the development of grapevine leaves. It's going from young leaves on the left to old leaves on the right, and so it's going to young now, back to old. And this is just to show you, you can see at a glance really easily using very simple statistics the types of ways that grapevines change their shape allometrically as they expand during development. Maybe your shapes don't have homologous features and you can't do a landmark analysis, and that's no problem either. There's something called an elliptical Fourier descriptor analysis, And this treats a shape as if it's a wave that goes back upon itself. So a shape is a closed contour, and just imagine if it was a wave that connected back up with itself, creating that closed contour. And when you treat a shape like this, you can do something called a Fourier analysis. And really briefly, a Fourier analysis, it takes any complicated wave and it can decompose it into an infinite series of sine waves, so that when you add up all the sine waves through constructive and destructive interference, it can recreate that wave. And so if you only take a finite number of elements in this series, you can create a really good approximation of shape. And again, you can do things like PCA analysis. And this was a work that I had done looking at the shapes of violins across their history. These were from photographs that were taken when about 10,000 different violins throughout history had been auctioned. And something I want to leave you with today is why we're interested in shape and morphology and form. The reason is that shapes contain information. When we're talking about biological shapes and organisms, we're talking about information in the form of genetics, environment, development. But there's also information in cultural objects, and you can see things, the cultural evolution and the information embedded through culture. And so in the case of violins, they started out with a particular shape at their very beginning, but this shape is very different from the shape of modern violins. And what this analysis showed was that you can see that it was Stradivari who first innovated this shape that we see in modern violins. And basically everybody was trying to copy Stradivari violins because they thought that they were superior. But actually, they're not necessarily the most superior violins, and actually shape has very little to do with their quality. But that really didn't matter. All that mattered is that everybody thought the shape contributed to a superior sound, and so everybody copied this form to create the modern shape of violins we see today. So that's just one example of how you can extract information content from shapes. So those are some examples of traditional morphometrics. It's an established field. They're very powerful methods. But we're going to talk about topology today. So let's do a bit of a primer into topological data analysis. And there is lots of jargon. And if you haven't really thought about topology before, within the next 10 or so minutes I'm going to talk about it. You're certainly not going to master it, and I am not a master in it at all myself. But what I hope to do today is just kind of let the topology wash over you. Topology is one of those things that I find is actually profoundly simple, and kind of like Mendelian genetics. It's very simple, but you kind of let it sit with you, and you keep on learning more and more things about it. And so that's how I think you should kind of treat topology. So I'll try to keep the jargon to a minimum, but this is only meant to be just a simple introduction. So what are topological features? These end up being blobs, holes, and voids. And there's this kind of dimensionality to topology. So blobs, topology is described with these Betti numbers. And zero-order Betti numbers are just blobs. And we're kind of looking at these ideal shapes here. One is just a point, a blob. Another is a circle. Another is a hollow sphere. Another is a hollow torus. This is a donut. It's a hollow donut. And then finally, we have a really wonky shape here called a Klein bottle. And this is something where you could walk along the handle through to the other side, through this wall of this vase. And so with a Klein bottle, if you were imagine walking up the center of this vase and through the handle and back out again, you could actually start on the inside of this object and end on the outside. And so it's kind of one of these Mobius strip-like things. When we're talking about a zero-order Betti number, all we're talking about are blobs. And each of these things is only one blob. It's only one contiguous thing. There's not multiple of these things. You can kind of think of them as little islands. They're not touching other things, and they're separate and contiguous unto themselves. If you go up one dimension, you get first-order Betti numbers, and these are holes. And so I think we can all agree that the blob has no hole. a circle has one hole. There is only one number on this chart that I really don't understand, and you can ask your favorite topologist why this is the case. But a hollow sphere does not actually have a hole. It has a void. We'll get to that in a second. Toruses have two holes. They have the center donut hole, and they also have this hole going around their hollow torus shape. And then the Klein bottles also have two holes. They have a hole for the handle, just kind of like the donut hole of the torus, and they also have a hole that counts for the hollowness of this vase. For voids, there is no void in the blob. There is no void in a circle. There is a void in a hollow sphere. There is one void in a hollow torus, and there's also one void in the Klein bottle, which is kind of like a vase. And these topological features keep on going up into n-dimensional space as well. And so these are kind of the features that we're dealing with. And so a common definition of topology that you'll see is that these are properties of space preserved under deformation, stretching, but not tearing things off and gluing them back together again. So topology is a very robust thing compared to shape. This is a very common example of that the coffee mug and the torus have the same topology with respect to the hole, which remains in the handle of the coffee mug. And even if you have a hollow torus, if that handle is hollow, that's being preserved as well. And yet the shape is completely changing. And so just think for a second to all the image analysis that you might be doing. So imagine watching little seedlings develop as they go through circumnutation and their cotyledons are flapping. Or imagine if you have some sort of video on a bunch of baboons and all their limbs are going all over the place and they're running and walking. The shapes of what I've just described have completely changed dramatically. But the topology did not change at all unless you tear off and glue back a limb or a branch of something. That would change the topological features, but not the shape. So that's a little bit of an intro into topology. And now how can we use this for data analysis? And to get to that point, you can think of networks as topological structures. And these are called simplicial complexes. But all it is a network. Another word for it is a graph. Except when you're talking about networks and topology, they're very concerned about dimensionality. And so one node is a vertex, two nodes are an edge, three are a triangle, and four is a tetrahedron. And so kind of just like the coffee mug and the torus, the pink and green shapes here both have the same topological features with respect to a hole. And you can see there's vertices here, triangles, and even tetrahedrons. So you can think of networks in a topological sense. And the reason that's important is because when we apply topological data analysis to the real world, the whole crux of it is to convert your data into a network. And so when I say your data, what I mean is any data set in which the distance of every one of your samples with every other sample is known, a metric set. And you can think of all sorts of examples in biology where we have this, where we have a pairwise distance matrix of everything to every other thing. As long as you know the distance of everything to another, you can do topological data analysis, and you do that by creating a network. And so if you have example data like this, to create a network, we need to describe a criterion where we make edges. And so we're going to use the distance between objects as a criterion, and we need some sort of function to describe that. So where after a minimum value of a function, we create an edge. So let's just use radius. And if we choose a particular radius, we just say we create an edge at every time the radii touch each other. And so you can see you start with all these independent islands, little blobs, which is zero order topology. But at a certain value of our radius, they end up making little islands. And so we've converted our data now to a network. But you might say, well, it was really arbitrary that we chose a radius value of this particular value. What if we had chose a different value? And that's exactly the case. If you had chosen a different radius value, um, you would have two different networks here. And so if we just increase the radius a little, we start seeing a larger island here. We get a local circle here. And so it's really arbitrary about how we create our network. And so one of the most widely used and powerful methods of topological data analysis is called persistent homology. What persistent homology does is, rather than choose a function value, it looks at all the function values, and it looks at all the networks, a continuum of networks, if we look at all the values and all the networks we can create. So we go along this function radius, which is also called a filtration, and we look at all the values, and as we go across it, you can see that the network changes dramatically. And this is how topology looks at features across scale. And so you can see you get local things like this little circle here, but eventually you get to a point where you start seeing this overall macro structure of a big ring, and this persists for a while. And then eventually you only have a blob. And so one way of thinking of persistent homology is you're looking at the shape and structure of your data at different resolutions, zoomed in and zoomed out. And so that's where we're looking at all the shape features globally, because we're looking at all the samples in our data set, and we're looking at them across scale as well, from the micro to the macro. We record the topological features, let's say in this case a blob, in something called a persistence diagram. And so it's a little hard to see here, but there's these little points in this diagram. And what these are is, for each of these features, the blobs are born at a particular function value of radius, which is the x-axis, and then they die at another particular value of the function. And when they die, that's when two things that were separate, for example, these two islands here, one of them dies when they merge into a bigger island. And for example, each of these little things in this circle, for example, each of them dies when they become a larger circle. And one of them dies, and one of them persists and keeps on going on. And it was the one who had persisted the longest that ends up persisting after one of them dies when they merge. And so what we're doing here is we are recording the birth and death of these features in a persistence diagram. So this is the whole crux of the thing. So let me go over this again in another diagram. We're dealing with networks here. We're dealing with single nodes, edges, triangles, and tetrahedrons. And we're starting with a pairwise distance matrix in which the distance of all points to each other are known. We take a function so that we determine where we're putting the edges, and then we end up with a network that we're using to do our analysis. And we do this over a continuum of values of the function or the filtration. And another way to record the data besides a persistence diagram is a persistence barcode, which is this thing here. And we're looking at zero order features, which are blobs. And you can see for a particular function value here, all these blobs that are here are all born at this one time. But then you can see as they start to merge with each other, a lot of them are dying here by the time we get to this one here. And then eventually, only one of these features persists in the final network that we create over the values of this function. We could have also looked at first order homology, which again is just loops. And you can see here at the beginning that there are no loops because there's only blobs. It takes a little while that we see our first loop here at this point in the structure. And then similar to the blobs, we record the birth and the death of all these loops. And again, we only have one loop that persists. So we're looking at topological features, blobs, holes, and with respect to our network, which is our data. And all our data is the known distance of all of our data points to each other. And we record topological features across a function. So that's what topological data analysis is. What is it good for? Morphometric methods work just great for 2D data, and you'll see that topology works for 2D data too. But we really need something different than traditional morphometrics if we're looking at 3D or even 4D structures. 4D structures are just 3D over time. Something that we can apply without any ad hoc analysis of us picking out our favorite traits. That if we just give you an X-ray CT scan, that you can come back with information content about that object, knowing nothing about it. So we're hoping that topology is a really promising method for X-ray CT data. What is X-ray CT data? What this is you take sequential 2D radiographs at multiple different angles, and from these you can create a 3D structure, which is computed tomography. So tomography is just taking slices of something, and you can create these beautiful 3D models. And let me just take you through some examples of where X-ray CT scans are just well suited for plant morphology. These are some sorghum panicles that I collected up in Nebraska of a variety of different sorghum varieties. And this is a scan that you can see that using an X-ray scan, there's no occlusion, and you can see the beautiful branching architecture of a sorghum panicle. This is some data from UC Riverside. This is showing how you can use X-ray CT scans to see the patterns and structure of damage in avocado. We're working with Jacob Landis and Daniel Koenig to look at the structure of barley panicles. These resulted from a composite crossing scheme at UC Davis. It's a long-term evolution experiment. This is a beautiful scan from Amy Litt, who's looking at manzanita, and you can see how well it picks up on these beautiful trichomes. This is just a scan of a maize plant and from Carolyn Rasmussen, and you can see the immature tassel in the middle of all the sheaves of the leaves of the stalk. And then my favorite, I work on grapevine. X-ray CT can allow you, for example, to see the structure of these buds where in grapevine, a lot of the organs for the shoot for the next year are already patterned in these buds. And you can see these secondary and tertiary meristems here, which are actually have pre-patterned organs for the year after next and the year after next. So you have 2 or 3 years growth in a little grape bud, but these are things you can see with X-ray CT. So that is what topological data analysis is useful for. We have this high dimensional data that has exquisite detail from small scale, microscales to the overall organismal level. We really don't have a good way to just quantify it in its totality for the information that it contains. To be using this to answer hypotheses or to be comparing all structures, patterns, and shapes against each other. So now I want to show you how we can use topological data analysis to study plant morphology. And I just kind of want to run really quickly through the various functions that we can use to describe morphology. And I'll give you a lot of different 2D examples with shapes, local features like serrations, and first order things like loops and shovelnomics data. And then I want to talk to you a little bit about 3D, about how we can use topological data analysis to measure branching architectures. And then finally, another type of topological data analysis called mapper, which converts morphology to a graph, and you can go back and forth between your actual data and the graph. And so you saw at the beginning how morphometrics is a powerful technique, but it does have some caveats. So landmark analysis fails if you don't have landmarks. So if you start looking at all the leaves across the land plants, the simple leaves, the compound leaves, the leaves with different numbers of lobes, you don't have any consistent homologous features that you can use as landmarks. Elliptical Fourier descriptors also fail because sometimes leaves have differing numbers of lobes, and that really throws off an analysis that's based on waves. So we developed a topological method, and this method treats a shape as a 2D point cloud where each pixel is a point. And then we're applying a Gaussian density estimator, and this is an estimator that tries to estimate the neighboring pixel density of each pixel. And so pixels, for example, that are in serrations or lobes, they tend to have a higher density of pixels around them, Whereas pixels that are on more straight lines, for example, they have fewer neighbors. And so this is just a function going from the most dense in red to the least dense in blue. And Mao made these beautiful visualizations. So she put it on its side and created this kind of topography to show you that the most dense things are kind of like mountain peaks in red, going down to the lower valleys in blue. And so Mao developed this function of density so that she could apply topological data analysis to shape. And she made this beautiful animation to show this. And so the function here is density, and we're going to start at the most dense, and we're going to record the number of blobs, the number of islands. And so you can see as you're proceeding down the function, you're recording the birth and death of the islands as they emerge and they merge with other ones. Each of these bars corresponds to a blob that somewhere along the way it was born and then it died, and then eventually only one of the blobs persists. And you can use this method where other morphometric methods fail, comparing simple to compound leaves to highly lobed leaves with different numbers of lobes. And this paper was just accepted in Frontiers, but I just want to mention that we deposited this data long ago publicly. You can download over 180,000 outlines of leaves. This is at Figshare. You can just search for leaf shape in Figshare and you'll probably find it. And these are leaves from over 140 plant families from 75 sites around the world. And it represents a lot of data from the community that was published and unpublished. A lot of this is paleoclimatic data that comes from Wilf, Royer, and Pepe. And a lot of the other data comes from all the collaborations over the years with a number of people looking at the genetics and ecology of leaf shape and all these different groups. So all these people contributed leaves to this project, and you can download them and analyze them yourself now. And it was, I know this is such a simple thing, but this allowed us to do a principal component analysis, which is essentially a leaf morphospace, a morphospace of all the shapes of the land plants, which is a simple thing, but a very gratifying thing to finally see the shape space of land plants. What if you don't want to look at all shape? What if you want to isolate shape features? So what if you want to look at serrations? You can use a very bad Fourier approximation of a leaf so that it's going in between the serrations, as you can see here. And then you can create a sign distance function which has positive and negative values of each pixel from this shape approximation. And then you look at these islands going across the function, and you record how many islands there are at each level of the function. And that picks up on serrations. What if you're looking at data like shovelomics data of a 2D image of a root? For that, you can create dilation, where you basically keep on dilating the roots, and you're recording the number of loops in this curve. And really quickly, this is comparing a parent tomato with a near isogenic line that has a small bit of genetic material from a desert relative. So these are lines that are very subtly different from each other. But if you plot out these curves for each replicate, for the serrations here and for the roots here, you can see that there are clear differences. So these are methods that pick up on very complicated features and can be used to distinguish genotypes, environments, development, and so on. These methods can be used to pick up on branching architecture, which is one of the really powerful aspects of topology for plant biology. Morphometric methods pick up on shape, but plants are generally not shapes. You can use shape for leaf shape, for seed shape, petal shape, things like that. But at the end of the day, plants are branching architectures. So how can we create a function that picks up on the topology of branches? Mao created this interesting method that is geodesic distance. Geodesic distance is the curved distance along an object. And here we're looking at the curved distance of any pixel to the base of this branch. And this is a very simple example, but you can see how it picks up on branching. And this is a more complicated example going where each voxel, each 3D pixel, the curved distance to the base of this grape cluster. So all these are going for each voxel down to the base. And as we do this, little islands are born and die, and this creates this persistence barcode where each of these bars corresponds to a feature. And eventually, only one survives. And actually, in this case, you can see two survive because it's a little cut off here, but you can see there's two branches that were kind of separated from each other at the base. Mao created this beautiful video that I think really shows how this works to pick up the branching architecture. So again, we have our 3D shape, a grape cluster, and Mao has colored each voxel by its geodesic distance from red to blue. And what you're going to see is we're going to traverse this filtration, this function, going from the most distant to the least distant voxels. And as you do that, little branch islands on branchlets merge on bigger branches, and you create this beautiful persistence barcode. We talked about how we could apply various filtrations or functions to describe the topology of plants on the nature of things in the American Journal of Botany. It's called the Persistent Homology and the Branching Topologies of Plants, so you can read that if you like. So why are, I kind of glossed over this, but why are we recording these persistent diagrams and barcodes? Well, what's the whole point of this exercise? And you can actually calculate the overall similarity and distance of any barcode or diagram to another. And this is something called a bottleneck distance. And I didn't get into it today, but just know that the whole point of this is that we're creating a distance matrix of the overall morphology captured with this topology using this method bottleneck distance. And you can use this, for example, to do a discriminant analysis on different grape clusters from different vines, for example. And so that's where we're going with this. It ultimately quantifies shape by creating an overall metric of the distance of any shape to another. So that's really powerful, that we can create these persistence diagrams, these persistence barcodes, and then we can calculate the overall distance of any object to another. But there's a problem, and that is that once you have a beautiful persistence barcode, you cannot go back to the original object. There is no correspondence back and forth between a persistence diagram or barcode with what you originally measured. And so that's a bit of a problem, both for interpreting what a persistence barcode or diagram is, and also creating a correspondence at a level other than topology, so that we can compare things based on kind of what we know and care about most. So there's another algorithm called Mapper. And let me talk to you a little bit about Mapper because it bypasses this problem. What Mapper does is, again, it's taking a function of data where you know the distance of any point to any other. And so the function here is really simple. It's just the y-axis value from yellow to blue, you can see here. And then the other part of Mapper is you create these intervals. And these are called covers. And the intervals must overlap with each other a little bit. And you can see what we're showing here is that each of the covers we're showing contains a set of different points. And some of these points overlap with each other. And from this overlap and these different intervals, you can create a graph where each of the nodes corresponds to sets of points, and the edges correspond to shared points, which corresponds to the overlap between the different intervals. Some of the points, when they're shared, are actually only shared with one interval but not another. So for example, these points here, which corresponds right here, are only shared with this interval between these two. But this interval here in the middle shares these points with this interval and those points with this interval, which corresponds to this. And maybe the best example for this was the original publication by Carlson, but it shows a hand, and I think this is a really intuitive example. If you start with a point cloud of a hand, you create these intervals that are overlapping with each other. I hope you can see that. But from this, each of the nodes corresponds to different sets. These three fingertips, for example, here. but they are shared with the other intervals, which are the edges, but they are still spatially distinct things in topology. Each of these is spatially separated blobs. And so this is how you get from raw data to a graph. And you can see there's a direct correspondence between the two. So let's look at this for a plant example. This is, again, a barley from Jacob Landis and Dan Koenig. And we apply a function, in this case, just the distance along the base to the tip. And we create these intervals. And you can see when the function corresponds to how we think about a panicle, for example, the orientation from base to tip, the graphical representation actually kind of corresponds to how we think about plant structure. You can use less intuitive functions like X-ray density, for example. So here we have the most dense of the seeds and we have less dense of the rachis. And you can use this too to create graphs which represent these structures. We're working on creating a very interactive way that you can actually see how different nodes and edges correspond to your original data. This is all the work of Binchel Ethan, who has been implementing this for visualization purposes. You may say that these intervals are a little arbitrary, that you could select different quartiles or different amounts of overlap, and you'd be right. But there is something called a Reeb graph, and this is kind of the limit of these intervals if they became infinitesimally small. So you can create a graph that is actually taking the limit of these intervals as they become smaller and smaller. And in this ideal example, this is a torus. The function is these level sets, which are going along its length. And you can take their limit to where they become infinitely small and get this graph. And so ultimately, that's where we're going to create these idealized graphs. But I hope you saw today that topology is a really powerful way to measure shape, structure, patterns. And in fact, that's its main interest at the moment. The reason people are interested in topology is that they want to use it in a data science way to look at the shape of data. And it just so happens that there are pure shapes in nature. And so this is why it's an idealized method to look at these things. It's very powerful because you don't have to come to this analysis with any assumptions. You can use functions like X-ray density and not make any assumptions about the shape at all or care anything about why you would be interested in a shape for a particular hypothesis. You can just extract the information content. And so beyond plants, we're looking to collaborate with the MSU Museum, which has natural history and anthropology samples, as well as the Art Museum at MSU, the Broad. And there's just these beautiful shapes of animals, inorganic matter, plants, that we have access to. And just imagine if we started scanning all the things, if we started looking at all the shapes that are out there, quantifying them from a data science, image analysis type of perspective. All these shapes have information. For biology, they contain information about genetics, environment, development, evolution. For anthropology, archaeology, like the violin example I gave, there's examples of cultural evolution and information that's transmitted culturally. And then for material sciences, there's information about structure. And so I think we're really poised that we have the right technology, like X-ray CT scans, with a known way to analyze shape and form through topology, to really make the study of shape and morphology a science into itself for once. And so that thank you. I know it's awkward that this is a pre-recording and I'm not here to answer any questions, but feel free, please, to at me at Morphology Lab on Twitter. And once I land, I'll be sure to get back to you. I hope to see some of you tonight. And I just wanted to thank all the organizers, especially Mitchell. I know it was an amazing symposium. I'm really sorry I missed it, but thank you for listening, and I hope to see you soon. Thanks.